Today we'll walk through the steps to properly calibrate a pH electrode, test different solutions, and understand how buffers work.
First, we carefully remove the electrode from its storage solution. Keeping it moist is essential for accurate readings. Next, we rinse it with deionized water to remove any residue before calibration.
We'll use a 2-point calibration method. We start by placing the electrode in a pH 4 buffer solution, entering 4 into the meter and rinsing the electrode. Then we repeat the process with a pH 7 buffer solution. Once completed, our meter is calibrated and ready for testing.
But what is buffer? And why it's important? A buffer solution resists changes in pH when acids and bases are added. It contains a weak acid and its conjugate base, or a weak base and its conjugate acid. When an acid is added, the conjugate base reacts with excess H+ ion to prevent a pH drop. If a base is added, the conjugate acid donates H+ to neutralize the OH- ion. This helps maintain equilibrium in the solution.
Now let's test the pH of different solutions. We start by rinsing the electrode with deionized water. Our first test is apple juice. We place the electrode in the solution and record the pH, then rinse. We repeat this process for orange juice, acetic acid, and hydrochloric acid, rinsing the electrode in between each test to prevent contamination.
Now let's analyze the differences between strong and weak acids. Strong acids, like HCl, fully dissociate in solution, releasing all their protons. Weak acids, like acetic acid, only partially dissociate, establishing an equilibrium between the acid and its conjugate base. This difference in proton dissociation affects pH stability. To minimize pH fluctuations, buffers are used to stabilize the environment.
In this experiment, we calibrated a pH electrode, tested different solutions, and explored how buffers maintain pH balance. Understanding these concepts is essential in chemistry, biology, and environmental science. Thanks for watching.